My name is Frank Nichols. I'm a faculty member in the Division of Periodontology, and I've been here for many years, worked with a lot of different people in terms of curriculum development. One of the individuals I worked with extensively, who's now retired, is Clarence Trummell, who I have to acknowledge here because of his contributions to the educational programs in the periodontics curriculum. But he and I had worked quite closely over many years, and that's why I've included him here on the title page. Now, what I'm going to talk about today are some features of periodontal abscesses. But before we can discuss these acute kind of periodontal diseases, I need to review some basic information about periodontal structures, the microbiology of chronic periodontal diseases, and and also how these diseases occur, so that you understand the baseline kind of predisposing conditions. So to start with, let's just review the definition of the periodontium. It's basically the tissues that surround and support the teeth. There are three basic components of the periodontium: the alveolar bone, which is the jawbone that specifically supports the teeth, is not the entire jawbone. The periodontal ligament, which runs from the jawbone, the alveolar bone, to the tooth, is the ligamentous structure called the periodontal ligament. And within, or the structure on the tooth that's actually attached to the periodontal ligament, is the cementum, which lines the root. The fourth component of the periodontium is called the gingiva, and that represents the soft tissue that surrounds the teeth that you can see in your mouth. Now, these structures can be depicted on a cartoon kind of a structure here. Imagine if you took a slice through a lower central incisor and turned it on its side and then drew out what the structures represent. You can see on the upper part of the slide the thickness of the enamel. It's depicted as that narrow kind of a zone that extends around the crown of the tooth. Underlying that is the dentin, and the central part of the root extending down to the apex is the pulp chamber. Now, what I want to point out to you is where you can see the gingiva actually sitting on the tooth or adjacent to the tooth. This is called the free gingival margin, and that is the structure that begins the gingiva, and it extends down apically for a distance of about 2 to 3 millimeters. Now, the important point about the gingiva is that the free gingiva does not actually physically attach to the tooth. There's a crevice that exists between the tooth crown and the free gingiva. There's a sulcus of about 1 to 2 millimeters in depth, and this is normal. So at the very base of that sulcus, though, there is a zone of what's called junctional epithelium, which actually physically attaches the gingiva through this epithelium to the tooth, and you see it depicted on this crown as that kind of pink-purple zone that extends around the crown of the tooth. That represents the first part of the attachment apparatus to the tooth. From the line just below that pink zone, that actually represents where the enamel ends and the root begins. This is called the so-called cemento-enamel junction. That's where the cementum begins to line the root, and this is where the connective tissue fibers actually begin to attach either soft tissue or hard tissue to the tooth. Now, as we pass down along the root on the left-hand side of the cartoon, you can see that there is a zone, a crosshatch area. That actually represents the soft tissue of the gingiva. It's the connective tissue of the gingiva. And there is a zone of about 1 millimeter of that connective tissue attachment that represents actually physical collagenous fibers that are attached to cementum that hold the gingiva to the tooth. But apical to that 1 millimeter zone or so, there is what's called alveolar bone. It turns out that the alveolar bone typically is about 1.5 millimeters from the cemento-enamel junction in most folks, including almost all of you, I'm sure. The alveolar bone then is physically attached to the tooth through that ligament, and you can see the obliquely lining fibers that extend sort of apically as you pass or as you visualize between the alveolar bone part of this diagram and the tooth root. Those fibers represent actually the periodontal ligament. So this is a just brief introduction of the structures of the periodontium in cartoon fashion. Now, let's just refine these definitions a little bit better, or just reinforce them. The alveolar bone is that part of the jawbone that actually physically supports the tooth. It's not the entire jawbone. The periodontal ligament is what attaches the tooth to the bone, but it's via cementum on the tooth root, which is a very thin layer of hard tissue. It's about anywhere from 50 to 200 microns in thickness, typically, and it's where the periodontal ligament embeds into the tooth. The gingiva, on the other hand, is the soft tissue part of the periodontium, and this is the pink tissue that surrounds the teeth that you can see in your mouths. It acts as a barrier to prevent microorganisms from invading the underlying connective tissues, but it also functions as an important place for the host response to occur when bacterial plaque accumulates on the teeth, which is what we're going to get into in just a few moments. This is an expanded view of the structures of the attachment, and you can see the very top part of this cartoon. You can see the outline of where the free gingival margin would be, and it's sort of this rounded structure. And there's a crevice of only about 1 millimeter or so before the junction epithelium is depicted in that wedge-like gray structure between the enamel and the connective tissue. Where the junction epithelium ends is about where the cementum begins, and from the beginning of the cementum to the alveolar bone, as I said, is about 1.5 millimeters typically. And this zone is where the denogingival fibers insert into cementum and then extend out into the soft tissues of the gingiva. Further apically, though, where the alveolar bone exists, the fibers inserting in cementum extend out to the alveolar bone, and that actually is what physically holds the tooth into the socket. Now, if you look in your mouth, you will see this zone of pink gingiva that surrounds the teeth. It extends for a width of about 2 to 3 millimeters from the free gingival margin, and you can see that there's a kind of an irregular junction between the very pink gingiva and the more reddish kind of oral mucosa. The difference between the gingiva and the oral mucosa, in addition to the color difference, is that the gingiva is very firmly bound down. You can't move it. If you put your finger on it and try to move it, it won't move. But the reddish oral mucosa, if you pull on your cheek or your lip and you look, you'll see that oral mucosa can actually physically move. So it's not bound down with the same kind of tenacity as the gingiva. It has much higher content of elastic tissue and much lower content of collagen. Now, the primary issue with periodontal diseases is that there is this problem of bacterial plaque accumulation around the teeth, which elicits a host response. Basically, it's a chronic inflammatory response that occurs in the adjacent tissues, and the chronic inflammatory response occurs primarily in the gingiva, and particularly in the tissue of the gingiva that surrounds that crevice. So it can involve both the junction epithelium and the connective tissue adjacent to the sulcus. Bacterial plaques are the kinds of bacterial accumulations that cause periodontal disease, and these are aggregates of bacteria that are tenaciously adhered to the tooth, and they can't be removed with, say, a spray from a water pick or a syringe or something like that. The bacterial plaque is very tenaciously absorbed to the tooth surface. It has to be mechanically disrupted, say by a toothbrush or floss, in order to remove it. The clinical definition of plaque is a firmly adherent film that cannot be removed by vigorous lavage with a spray from a water pick, for example. But it can be removed with mechanical means. And we'll get into some of the details about these plaques in just a minute. But let's define right now the differences between the different kinds of plaques that can occur on a tooth. There is both supragingival and subgingival plaque that can occur. The demarcation between supra and subgingival plaque occurs at the free gingival margin, which is where you see the gingiva sitting on the tooth. So within the crevice, there is a subgingival plaque. But it's more complex than just simply a plaque, an aggregate of nonspecific organisms. It has both a firmly adherent component and a loosely adherent component, meaning some of the bacteria are firmly adhered to the tooth structures within the sulcus, and others that are layered on top are much more loosely adhering, and they can be rinsed out they can be basically irrigated out to some extent. Now, in addition to bacterial plaque that can accumulate on teeth, there are other kinds of films that can occur. Materia alba is basically food deposits that are kind of whitish in color. They can contain bacteria. The primary difference between materia alba and dental plaque is that materia alba can be rinsed away with a syringe or a spray like a water pick, but bacterial plaque can't. Pellicle is the adsorbed organic layer that forms on a tooth soon after it's cleaned, either mechanically with a toothbrush or by a professional individual that uses a prophylaxis kind of an instrument. The pellicle is basically salivary components that adsorb to the mineral of the crown of a tooth, and they form the underlying layer to which the organisms initially colonize to start to form the bacterial plaque that forms on teeth. Stains, on the other hand, are usually extrinsic materials that accumulate on teeth simply because there are pigments in food or drinks that we consume that will stain the teeth. Stains and pellicle are not pathological. They don't elicit any kind of a host response. They don't cause inflammatory disease or any kind of a problem for the host. In contrast, calculus basically represents mineralized bacterial plaque. It can also occur as supragingival or subgingival calculus. The supragingival calculus usually forms more significantly where salivary gland ducts release saliva. So on the lingual aspects of lower anterior teeth is a very common place for supragingival calculus to form. On the other hand, subgingival calculus can form anywhere where there's a sulcus, even a sulcus that's only 1 millimeter deep. It can form subgingivally, and I'll explain more about how that happens in a little bit. Going back to what plaque represents and how it looks, the picture on the left-hand side depicts clean tooth surfaces. They look very clean. The gingiva that's adjacent to the tooth surface, that's the free gingival margin there. It looks nice and pink. It's knife-edged. It's healthy. There's no obvious clinical sign of any kind of inflammatory changes. On the right-hand side, you can see that there's this whitish material film that has accumulated right at the gingival margin, and it has some degree of thickness. But also notice that the gingival margin adjacent to that plaque also has a more reddish kind of an appearance than the more apical zone of the pink gingiva. That represents the clinical kind of inflammatory response that's typically seen with initial kind of plaque formation. So this is the kind of plaque that we're talking about that cannot be removed unless you use a toothbrush or floss. When the teeth are cleaned with a toothbrush, basically there are no bacteria on the teeth. But within a relatively short period of time, minutes to hours, there's an accumulation of the pellicle, and that then is followed by the accumulation of organisms, which can be found just as individual organisms that adhere to some of those proteins that are adsorbed to the mineral of the tooth. This is a slide showing the kinds of organisms, the morphotypes of organisms that accumulate on a tooth surface within 8 hours after cleaning the tooth. And you can see there's this dome-shaped kind of a colony of organisms and some just individual organisms sort of scattered about. This is a fairly thin form of plaque, and it would be so slight that it wouldn't be visible to the naked eye. Within 24 hours, though, you can see that there is a much thicker zone of organisms, and there's a hierarchical kind of a distribution of the organisms. On the left-hand side, those organisms that are adsorbed to the tooth, you can see they're oriented in sort of a parallel direction, parallel with one another in a more organized kind of a way. And then as you pass over to the right-hand side, you can see it's less well organized. There are different kinds of morphologies to the organism. Some are dense, some are light. They're mixed in some cells from the host, for example. So this is the way that plaque continues to form as a more diverse kind of a constellation of organisms. And then after 3 days, it becomes a mass of densely packed organisms against the tooth. And at this point, you can definitely see a plaque mass on a tooth. It becomes visible to the naked eye. This is supragingival plaque. So the way it looks clinically, there can be a very thin film of plaque. For example, this is an individual that has cleaned relatively recently. And you can see on the left-hand side, there doesn't appear to be any visible plaque on the teeth. But if you stain with a red kind of erythrocin stain that we have available, you can see that along the gingival margin there is a zone of a couple of millimeters of this accumulated red material, which actually represents the stained organisms that accumulate over a relatively short period of time. This is probably a 24-hour accumulation of plaque, but it shows exactly where the plaque accumulates relative to the gingiva. It forms right at the gingival crevice, and it's the least effectively removed plaque with mechanical measures. This is where we work with patients and trying to convince them to change their technique so that they're more effective in cleaning their teeth right around the gingival margin. In contrast to the plaque, which is soft microbes, microorganisms, calculus is hard. It's mineralized, and it's mineralized plaque, basically. And it comes in two forms, as I mentioned, supragingival and subgingival. You can see on the left-hand side, the supragingival plaque is stained with pigment, probably from coffee or tea, but it is gross in its amount. And you can also appreciate the severity of the gingival inflammation on at the marginal gingival level, and even on the lower anterior teeth, the facial gingiva is so inflamed it actually looks like it's retractable from the teeth. On the right-hand side, that frame shows the position of a spicule or a ledge of subgingival calculus relative to the position of the sulcular epithelium. The tissue is actually physically detached, but you can appreciate where the free gingival margin is at the about the top third of the slide, and then as you pass down to the mid portion of the slide, you can see the calculus and where it would sit adjacent to the sulcular epithelium, and then further apically is where the junction epithelium would have been attached to the tooth. So this is subgingival calculus, which can form virtually anywhere in the mouth, and it doesn't matter how deep the sulcuses are. It can form, and sulcuses are very shallow or very deep. It basically represents either supragingival or subgingival calculus represents bacteria that have mineralized on the tooth surface. Basically, the bacteria are born, live, and die within a period of about four hours or so. When they die, they can mineralize, and this is how you develop these layers of subgingival or supragingival calculus on the teeth. It can be removed, but not with a toothbrush or floss. It has to be physically scaled off with steel instruments. Now, in terms of the way gingivitis occurs relative to supragingival plaque accumulation, this slide is depicting what's called experimental gingivitis. And the yellow dots on the left-hand side show the increase in the thickness of the supragingival plaque measured with a qualitative kind of an index that we use. And so this happens from day 0. You can see that there is this increased thickness of the supragingival plaque, but this is occurring right at the gingival margin. It's measured only at the gingival margin, not over the rest of the tooth surface. In comparison to the increased plaque thickness, you can also see the red dots showing the increase in the visible kind of redness in the gingiva or the tendency for bleeding on provocation. This is called the gingival index, and it measures the severity of the gingival inflammation. And so it's pretty clear that the thickness of the supragingival plaque, as it increases, you see this increased tendency for gingival inflammation to exist in the adjacent tissue. This is extended out for a period of 21 days or 3 weeks, at which time oral hygiene measures were reinstituted. And you can see that the plaque accumulation and the gingival inflammation are reversible. They go back to the baseline levels with just simple reinstitution of oral hygiene measures. That would suggest that the supragingival plaque is the cause of the gingivitis, but it's a little bit more complex than that. Because if we look at the subgingival microorganisms that accumulate, there is a different kind of a flora that accumulates, and the establishment of the organisms in different kinds of morphotypes changes depending on the time or the duration of the plaque accumulation. So what this slide is showing is that in the gray striped area, there is this first accumulation in the subgingival area of coxoid cells and small filaments. Then after about 7 days, 5 to 7 days, there's an increased percentage of what are called filamentous kind of organisms, larger kind of rod-like organisms that can be curved and straight and so on. And then following that, around 10 days or so, there's a significant increase in the morphological organisms that can move. These would be spirochetes and vibrios. Vibrios are motile rods that can swim around. When these organisms appear in the plaque, this is when the gingivitis is most significant, and it increases in severity for about another week and a half or so. And you can see that depicted at the top of the slide as the wedge, the black wedge where the gingivitis severity increases. So to reiterate, it is felt, I think, by the experts in the field that it is the shifts in the subgingival flora that correlate best with the increased severity of gingivitis that has been shown to occur as plaque accumulation occurs supragingivally around the teeth. So in essence, the supragingival plaque is not the primary cause for the increased severity of gingival inflammation. It's the shifts in the subgingival flora. So we need to review the information about what types of organisms actually accumulate. But before we do that, I just want to reinforce for you the factors that determine the severity of gingivitis related to plaque, and that is the duration of the plaque accumulation. The longer the plaque is on the teeth, the more severe the inflammation is, as you saw on that diagram in a previous slide. The quantity of the plaque, that is the thickness of the plaque at the pregingival margin, is also critical for determining the shift in the subgingival flora, which ultimately cause more severe gingival inflammation. That basically is represented by the microbial composition of the subgingival plaque, changes that occur. And then superimposed on all this is the possibility that the host response may be altered. For example, some individuals have immunodeficiency issues that can severely affect the way microbial surveillance occurs in the sulcus, and they are much more prone to periodontal diseases. And we'll review a few of the types of diseases later on that can predispose for more severe periodontitis. This slide shows a cartoon basically of the subgingival crevice and how the organisms accumulate in the crevice. You see the upper blue arrow indicates the pregingival margin, the lower blue indicates the alveolar crest, the alveolar bone crest. The left-hand side is where the tooth is, and the surface of the tooth is colonized in the sulcus by these parallel-oriented organisms that would represent the firmly adherent subgingival plaque that I mentioned earlier. On the surface of those well-organized, firmly adherent organisms are a group of more loosely adherent organisms that can include those motile organisms that we talked about earlier. And then along the surface of the sulcular epithelium, you can see on the more right-hand side of the sulcus there are organisms that actually are lining the epithelial layer. And I'll describe a little bit more about those in just a moment. The important point here is that there is a change in the composition of the subgingival plaque as we move from the surface of the tooth to the sulcular epithelium. And but regardless of the change in the composition of the types of organisms, most of these organisms are strictly anaerobic organisms. They do not grow in the presence of oxygen, and they are gram-negative organisms, which makes them quite distinct from supragingival plaque. Now, this slide shows the groups of organisms that are known to grow together in what are called clusters. The organisms that are encircled in different colors on the left-hand side of the slide represent those organisms that are found in the firmly adherent plaque. They're the first ones that form on the root in the sulcus. What this is showing is that, for example, in the upper pink circle, the Villanella parvula, when that is found in this subgingival plaque, you're much more likely to find Actinobacillus odontolyticus. The same applies to the yellow encircled organisms and the green ones, where if you find strep mitis in the yellow circle, for example, you're much more likely to find the other streps present. And in the lower green circle, Ichinella corrodens, if you find that in this firmly adherent layer, you're much more likely to find the various species of Capnocytophaga. These represent the first organisms that colonize the subgingival space, and they are the firmly adherent types of organisms. Now, following their accumulation on the tooth surface in the sulcus, this is all subgingival microbiology, there can be this intermediate cluster of organisms which are represented in the orange circles in the center of the slide. The center circle represents two basic groups of organisms that aggregate very, very strongly with each other. The upper organisms are Prevotella organisms that are represented in the Bacteroidetes phylum. The lower ones are Fusobacterium types of organisms from the Fusobacteria phylum. These organisms colonize very, very nicely together. They like to grow together. The outer circle, orange circle organisms also are associated, but not quite as strongly. And then these organisms, when they accumulate, they're more strongly associated with gingivitis expression. But on the right-hand side, you can see those organisms encircled in red. They represent three organisms that are associated most strongly with the destructive form of periodontal disease called chronic periodontitis. What happens in chronic periodontitis, you still have superimposed gingivitis present, but there's also present a destruction of bone and formation of a periodontal pocket through detachment of the soft tissue from the tooth. So there's loss of clinical attachment at the same time. The organisms in the red cluster are called Porphyromonas gingivalis, Tannerella forsythia, and Treponema denticola, and they are the organisms most strongly associated with expression of chronic periodontitis. Just to reinforce this idea of the aggregation of bacteria that occurs in subgingival plaque, here you see a large rod-like organism on the the left-hand side. This is Fusobacterium nucleatum, and to it is attached some of the Porphyromonas gingivalis organisms. They used to be called Bacteroides gingivalis, but now they've been renamed. And you can see these hair-like structures extending from the surface of the bacteria that are basically these what are called pili that the bacteria use to aggregate to one another, and also to aggregate to human tissues, cells, and tissues. And to some extent, these cell surface products are recognized by the host, and they can elicit inflammatory responses. So, for example, fimbriae, which make up the bulk of these adhesion types of proteins, are also recognized by the host through macrophage receptor recognition, and they promote chronic inflammatory responses. So this represents the type of aggregation that can occur and how it's easy to understand how these clusters can form in the subgingival plaque. Another organism that's unusual is one of the red cluster organisms, Porphyromonas gingivalis, and the reason why it's unusual is that this one can actually physically adhere to epithelial cells. So this is an example of a Porphyromonas gingivalis organism that has adhered to an epithelial cell in culture, and you can see the pseudopods of the epithelial cell extending up around each side of the Porphyromonas gingivalis. Now, one of the things about Porphyromonas gingivalis, it is a gram-negative anaerobic, strict anaerobic, pleomorphic kind of an organism. But if you look closely at the circumference of the bacteria, you can see these hair-like structures extending out. But just within that zone, you can see this railroad track kind of a structure. What that represents is the double membrane typical of all gram-negative organisms, and between each of the membranes is the periplasmic space, where lipopolysaccharide, for example, is produced. But what happens here is the epithelial cell engulfs this bacteria, and it ingests it basically and kills it. But here you can see that the bacteria is fully engulfed in the epithelial cell, and with this engulfment, there is activation of the epithelial cell to produce various kinds of factors that are antibacterial, which we don't have time to discuss. Anyway, these are the basic types of organisms that colonize the subgingival plaque and that are responsible for promoting, in those individuals who are susceptible, they're responsible for promoting the expression of chronic periodontitis. Now, in terms of the sequence of diseases that can occur after plaque accumulation exists on a tooth, we always will see gingivitis. Gingivitis is chronic inflammatory periodontal disease, but there is no destruction of the attachment or the supporting alveolar bone. Gingivitis will always occur when plaque accumulation exists on a tooth for an extended period of time. In addition to gingivitis, though, some individuals and some sites will show what's called destructive periodontitis, which is where there is destruction of the alveolar bone and the connective tissue attachment. And with that, there is a deepening of the sulcus, that crevice around the tooth, such that it can increase in depth up to basically the length of the root if it's a really severe kind of a problem. The point about all this is that you will never see periodontitis occurring without pre-existing gingivitis. Gingivitis always precedes the expression of periodontitis, and not all sites with gingivitis will ultimately develop periodontitis, only a small percentage. But it's enough that it keeps us busy in terms of our field. So periodontitis is really the most significant problem that we deal with in terms of destructive forms of periodontal disease. But both of these diseases, chronic gingivitis and chronic periodontitis, are chronic diseases. They are not acute. So these are the predisposing kind of diseases that have to exist in order to see the acute kind of periodontal diseases that we're going to talk about in a little bit. Now, just to show you what periodontitis, chronic periodontitis, can look like, here are a couple of slides that show what how we determine whether periodontitis exists, and it involves an instrument called a periodontal probe. And basically, it's a measuring instrument to see how deep the sulcus is around the teeth. And so, on the facial surface on the left-hand slide, you can see that the probe is inserted to some extent. It actually turns out to be around 3 millimeters based on the markings on the probe. But on the right-hand side, you can see that the probe extends much farther apically into the sulcus. This is what a periodontal pocket represents. It means that there has been loss of attachment, so there's a deepening of the sulcus. It doesn't appear as though the gingiva is inflamed, but if we were to look at it histologically, it would be very, very inflamed. But the point is that in periodontitis, the only way you can detect it legitimately is by using a probe. Now, these conditions, gingivitis and periodontitis, we're going to use as our baseline predisposing conditions now to discuss the acute kind of periodontal diseases.